The down position is if to hide the RBD and therefore evade the immune system. What causes it to turn up though? Like how does it go from down to up? Okay, hold on. Last thing, good luck. The life cycle of SARS-CoV-2, scientific version.
In our airways, the air we breathe comes into contact with a mixture of different types of healthy respiratory cells.
Respiratory?
Some
really?
respiratory cells have cilia, hair-like structures that move the mucus along. These ciliated cells in our airways are the main target for the SARS-CoV-2 proteins on it.
We know this. What was it? It was spike, membrane. That wasn't even it. It was SM and E. I don't remember what they stood for. Fuck me. Fuck me and fuck my ass. Yes! Yes! Hold on. No. Spike, envelope, and membrane are on the outside.
Spike, envelope, and membrane proteins. Inside the virus, an assembly of nucleocapsid proteins hold together its genetic material.
Cool.
A single strand of viral RNA.
And cringe.
The spike protein is responsible for making contact with the host cell and starting the infection.
Okay.
It's glycosylated, meaning it has a coat of sugars that hides it from the body's immune system.
That's crazy.
Many of the variants of SARS-CoV-2 are characterized by mutations on this spike protein that can influence its function, some making the virus more infectious. Many of these mutations are unique to each variant, but some, like D416G, are found on each of the variants of concern.
Okay.
When the virus closes in on a host cell, its spike proteins open up and bind to a receptor on the host cell called ACE2.
Open up. That's that up-down shit, right? That's what that is? Why does it open? Transition to up position is critical for RBD binding to ACE2. I wonder why. Whatever. Wait. I don't think we need to know this much though, but fuck it. Fuck it. Why the fuck not, dude? PPC motif. What does PPC mean? In SARS-CoV-2 spike protein. The PPC site. The proline site. Oh, is it just 3 amino acids? Aren't they supposed to be 4 at a time? Okay, whatever. Okay, different stages of coronavirus entry where host cellular proteases may activate different stages of coronavirus entry. Okay. B is schematic drawing of the three-dimensional structure of the coronavirus. S1, receptor binding subunit. S2, membrane fusion subunit. TM is the transmembrane anchor. IC is the intracellular tail. Schematic drawing of the 1D structure of the coronavirus. No, it's pro-protein convertase. Okay. Okay. The dynamic state of the RBD in coronavirus spikes may explain this paradox. The RBD in coronaviruses can be in either a standing up state, which enables receptor binding, or a lying down state, which does not bind to the host receptors. Cryo-EM studies have shown that the SARS-CoV spike, the RBD, is mostly in the standing up state. However, in SARS-CoV-2 spike, the RBD is mostly in the lying down state. Therefore, compared to SARS-CoV, although SARS-CoV-2 RBD has higher ACE2 binding affinity, it is less accessible, resulting in comparable or lower ACE2 binding affinity for the SARS-CoV-2 spike. So What makes it stand up? To maintain its high infectivity while keeping its RBD less accessible, SARS-CoV-2 relies on a second strategy, host protease activation. Host protease activation, protease activation, is a significant determinant of coronavirus infection and pathogenesis and a significant target for host immune surveillance and human intervention strategies. A combination of mutagenesis, protease inhibitors, and siRNA approaches. Here we show that Furin pre-activation enhances SARS-CoV-2 pseudovirus entry into different types of ACE2 expressing cell lines, including lung epithelial and lung fibroblast cell lines. We also show that cell surface protease, TMPRSS2, and lysosomal cathepsin activate SARS-CoV-2 virus. Comparison, SARS-CoV pseudovirus, but not Furin. Furin pre-activation allows SARS-CoV-2 to be less dependent on target cells, enhancing its entry into some target cells, particularly. Wait, so do we not know, or? Okay, how coronavirus entry is regulated by proteases. Protease activation of coronavirus spikes potentially leads to the final structural change of coronavirus S2 needed for membrane fusion. This process is irreversible and needs to be tightly regulated. Indeed, it has been shown that SARS-CoV-2 virus particles, many spike molecules have already undergone the final structure change. Hence, in principle, virus particles pre-activated by furin may have unchanged or reduced entry efficiency in some types of cells with high expressions of... Oh, so this up shit gets activated inside of the cell before it's spit out. That's what the Furin cleavage shit is doing? Imagine if that's what it's actually doing, and then I... Is that what it does? Is that what the Furin cleavage shit does? Is it turns the down to up before it ejects from the cell, or... Okay. For understanding clinical features of coronavirus disease, the hidden RBD can evade immune surveillance. When it says evade immune surveillance, can your immune system check for weird, like, organelles? Like, does your immune system look inside your cells? Because I'm trying to figure out how can a down RBD evade your immune system before it's left the cell. Because I imagine that so if I'm, okay. No, yes, no, yes, yes. Your cells have their own immune techniques, but immune cells aren't looking in. The hidden RBD can evade, potentially leading to insufficient immune response to prolonged recovery time. Granted, there are other immune, for example, some coronaviruses, importantly, viruses commonly hide their RBD or other critical parts of their spike proteins from host adaptive immune responses using two main strategies. First is conformational masking, where viruses conceal RBDs in locations like canyons or recessed pockets. The second is glycan shielding, where viruses conceal critical parts of their spike proteins behind glycan clusters. Our finding about the discrepancy in ACE2 binding affinity between SARS-CoV-2 RBD and spike, combined with other groups' observations of the lying down RBD and SARS-CoV-2 spikes, suggests that the hidden RBD contributes to the immune evasion of SARS-CoV-2 as one of the conformational masking strategies. Indeed, a recent study showed that SARS-CoV RBD induced mouse serum blood with high affinity but poorly. Result shows the immune surveillance recognizes hidden RBD less well than exposed RBD. However, hidden RBD may lead to poor recognition of the host receptor and inefficient entry into host cells. SARS-CoV-2 overcomes the problem by evolving an RBD with high ACE2 binding affinity and a furin motif that allows its spike to be pre-activated. The end result is that the overall entry efficiencies of SARS-CoV-2 and SARS-CoV pseudoviruses are comparable. Understanding the cell entry mechanism of SARS-CoV-2 can inform intervention strategies. The RBD is the most immunogenic region of the whole spike. Hence, the hidden RBD of SARS-CoV-2 presents a major challenge to both vaccination and antibody drug therapy due to the limited access of neutralizing antibodies to the target. Correspondingly, there are several approaches for intervention strategy. Okay. Okay. Answer. I need you to answer this question. I'm banning you. I need a clear answer for the MIW. So, at what stage in your body is the SARS-CoV-2 virus present with the RBD ever hidden? Because it sounds like the RBD would be activated by furin when it's created in the cell. And then I'm imagining once it goes out of your cell, it's always going around the body with the RBD activated. Would it not be? So where is it? How is it going through the body with the RBD not activated? You'll find the answer if you finish watching the video. Okay.
After the spike protein makes...
The RBD constantly switches between a standing up position for receptor binding and a lying down position. From this article, wait. Oh, so then, wait, so then... Cleavage doesn't turn it on. You were wrong when you said it. You lied to me. You lied. RBD is naturally always hidden. Cleavage allows it to stabilize the up position when it binds to ACE2 in the next target cell. But how can it bind to ACE2 in the down position? It's saying that it... Okay, let's see if this video tells us. This is an unnecessary level of depth. I don't think that we need this for this, even for this debate, but...
contact, furin cuts off the outer part of the spike protein, called the S1 domain. On the host skin on a host cell, its spike proteins open up and bind to a receptor on the host cell, called ACE2. After the spike protein makes contact, furin cuts off the outer part of the spike protein, called the S1 domain. This frees the inner core of the spike protein, called the S2 domain.
Okay, this is taking us back to the explanation that this guy said was wrong, though, the entire time. What the MIW was saying. Fuck, I just need to unlearn what I just spent the last, 15 minutes trying to... okay, never mind. All right. The life cycle of SARS-CoV-2 scientific version. We'll just throw this video here. Wait, we already opened this. Okay. Sorry, Jesus. All right, I'm watching this one more time, then we're moving on this debate. Jesus.
In our airways, the air we breathe comes into contact with a mixture of different types of healthy respiratory cells. Some respiratory cells have cilia, hair-like structures that move the mucus along. These ciliated cells in our airways are the main target for the SARS-CoV-2 coronavirus.
And the reason why they're the main target is because these cells tend to have that, the ACE2 gateway to bind that the SARS-CoV-2 binds to, right? They have that thing, the ACE2 guy. And ACE2 is a protein that's expressed on the outside of the cell, the cellular membrane. I don't know how you would say that scientifically, but
The virus itself is deceptively simple. It only consists of a few parts, but together these parts create a highly effective virus. It has an outer membrane with 3 types of proteins on it: spike, envelope, and membrane proteins. Inside the virus, an assembly of nucleocapsid proteins hold together its genetic material, a single strand of viral RNA. The spike protein is responsible for making contact with the host cell and starting the infection. It's glycosylated, meaning it has a coat of sugars that hides it from the body's immune system. Many of the variants of SARS-CoV-2 are characterized by mutations on this spike protein.
Okay, so it doesn't sound like this talks about that up-down switch as much, which maybe, I guess I'm not going to worry about understanding this, I guess. Like this transition to up position is critical for RBD binding to ACE2. I'm just never going to understand this. I don't know. Oh, yeah, I got it right now. All right. I explained it, stalk me. The spike protein exists in dynamic equilibrium between RBD up and RBD down. Flipping occurs. Why would it exist? Why would it keep going up and down? How does that benefit it? Then it seems like it would have equal chance to get taken up by a cell or recognized by the immune system. D614G mutation shifts equilibrium of up-down conformation towards up, which increases the likelihood of ACE2 binding. But it seems like that also increases the likelihood of it being detected by the immune system. It sounds like the whole meme is that this stays down, which makes it almost impossible for the immune system to detect, and then it flips up to do the binding. But I just don't know what causes the flip up.
Protein that can influence its function, some making the virus more infectious. Many of these mutations are unique to each variant, but some, like D416G, are found on each of the variants of concern. When the virus closes in on a host cell, its spike proteins open up and bind to a receptor on the host cell called ACE2.
Like, why does that happen?
Proteins open up, and when the virus closes in on a host cell, its spike proteins open up and bind to a receptor on the host cell.
Like this configuration here allows it to both be detected by the immune system and allows it to bind to ACE2. But the special meme with this coronavirus is that it's generally in the down position, which allows it to elude the immune system until it goes to bind. I just don't know why that happens.
Genetic code.
Wait, is this the glycans thing? Analysis of the S protein opening pathway reveals that glycans at N165 and N122 interfere with hydrogen bonds between the RBD and the N-terminal domain in the up state, while glycans at N165 and N343 can stabilize both the down and up states. In the Nature article, read the RBD section. This in this article? Control F, RBD. The three RBDs of the S trimer form the apex of the S protein, which samples two distinct conformations, up for a receptor accessible state and down for a receptor inaccessible state. In the post-fusion state, conformational changes lead to S1 subunit disengagement from S2, and likely its dissociation from S2, while S2 undergoes a cascade of refolding. This is the Furin cut. So what you kept saying in chat, okay, the one guy, the MIW, this is where, this is the Furin cleavage that's happening here. After it fuses in the post-fusion state, conformational changes lead to the S1 subunit disengagement. The S1 subunit is being cut by the Furin protease at this point, I think. And then likely dissociation from S2, while S2 undergoes a cascade of refolding events to form a stable and elongated trimer. The post I don't know where we don't need to know this. I don't need to know this. I don't even give a fuck. We do need to know? Shut the fuck up. We don't need to know. The NTD is formed mainly by 4 stacked beta sheets and a number of connecting flexible loops bearing several end-linked glycans. Whether the NTD plays any functional role in the SARS-CoV-2 entry remains unknown. Similar entities appear to facilitate the binding of sugar moieties. I don't know this, acting as attachment factors. The NTD of transmissible gastroenteritis virus and MERS-CoV may facilitate prefusion to post-fusion transition of the S protein. Something that may be functionally important, or at least the RBD. The RBD has 2 subdomains, a core structure formed by a 5-stranded anti-parallel beta sheet covered with short connecting alpha helices on both sides, and an extended loop named the receptor binding motif, which wraps around one edge of the core structure and makes all the contacts with ACE2. In the down conformation of the pre-fusion S trimer, a single RBD packs against the central helical bundle of S2 and the 2 other RBDs while leaning on CTD1, I don't know what that is, from the same protomer, and the NTD from a neighboring protomer. This configuration partially occludes the RBM, making it inaccessible to the receptor ACE2, so it can't bind in this state. When the RBD flips to the up conformation and fully exposes the RBM, I'm assuming RBM is receptor binding mechanism. That's what RBM stands for. When the RBD flips to the up conformation and fully exposes the RBM. Oh, I'm sorry, receptor binding motif, my bad. The adjacent CTD1 and NTD also shift away to accommodate the RBD movement. Carboxy terminal domains. Okay. The amino acid terminal, the amino terminal domain, the RBD, and two carboxy terminal domains, CTD1 and CTD2. Thus, the transition of the RBD from the down conformation to the up conformation is a critical step before the receptor can fully engage, and the 1 RBD up conformation appears to be a stable intermediate state. RBDs from other SARS-CoV-viruses can be categorized into two distinct groups, human ACE2 binders and non-human ACE2 binders. They are likely to fold similarly because of the high degree of sequence identity in the core subdomain, but variations in the ACE2 binding interface in the RBM of the non-human ACE2 binders can explain their inability to use human ACE2 as an entry receptor. Destiny, importantly, because immune cells require stronger binding, having this flipping helps it avoid strong binding to antigen receptors on immune cells, making the receptor. Show me that. You have to demonstrate that, because it makes it sound like when it's turned down, it sounds like it can't bind to ACE2 at all. You're making it sound like it can weakly bind, so it can go to ACE2, but not the immune system. That doesn't make any sense at all. If that was the case, it feels like viruses would just adapt to all weakly connect to cells, and then they would never be found by your immune system ever. But that doesn't sound like. You would have to show me that somewhere in a paper, because this is saying this configuration partially occludes the RBM, making it inaccessible to the receptor ACE2. Inaccessible to me doesn't sound like doesn't bind that often. That makes it sound like it is literally impossible to bind. It cannot bind in the down configuration. It has to be flipped up first. Importantly, the RBD is also the primary target of the neutralizing antibodies elicited by natural infection or vaccination. There are three major non-overlapping antigenic sites on the RBD. Most RBD-directed neutralizing antibodies, including REGN 10933, and C144, recognize the tip region of the RBM, and they often show remarkable potency blocking ACE2 engagement by direct competition. Another site lies on the exposed surface of the RBD when it is in the down configuration and is targeted by antibodies such as REGN 10987 and S309. The third site, often referred to as a cryptic supersite targeted by CR3022, is on the buried side of the RBD and is fully accessible only when the domain is in the up conformation. Together, these sites comprise the epitopes of majority of neutralizing antibodies to SARS-CoV-2, as discussed later. It's not going to say why it turns up or down. There's no way. We're not going to learn it. And it's not relevant. It doesn't matter. We're not going to know, and it doesn't matter. Structural element in S2 named the fusion peptide proximal region, or FPPR, abutting the opposite side of CTD1 from the RBD, appears to help clamp down the RBD and stabilize the closed conformation of the S trimer. Therefore, CTD1 appears to be a structural relay between the RBD and the FPPR. It senses the changes in the RBD and the FPPR, which are located on either side of the CTD1. CTD2 is formed by two stacked beta sheets containing 4 strands. In the other sheet, an interstrand loop contains the furin cleavage site at the S1, S2 boundary, and one strand is the N-terminal segment of S2. A structural element designated the 630 loop, and CTD2 is largely disordered in the S-trimer, and the 1 strand with disorder in the presence of the D614G mutation and plays an important role in stabilizing the S-trimer. S2 subunit. In the pre-fusion conformation, the S2 subunit adopts a conformation with most of the polypeptide chain packed around a 3-stranded coil formed by the central helices. Okay, I don't think this matters to us. The post-fusion S2 structure, one essential helix form an unusually long central 3-stranded the internal origin of the okay. ACE2 is an 805 amino acid carboxypeptidase that removes a single amino acid from the C terminus of its substrates. It consists of a single metallopeptidase domain, and the first half contains a blah, 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 blah. It's to convert angiotensin 1 and angiotensin 2 generated by renin and ACE into angiotensin 1 through 9, angiotensin 1 through 7, respectively. Like ACE2 receptors, the obligate receptors for several other coronaviruses and also proteases. Interestingly, the catalytic activity receptors neither is necessary for the receptor function, nor overlaps with the virus binding site. Consistently, small molecule inhibitors of the catalytic site of ACE2 do not block infection by SARS-CoV. Nonetheless, studies have shown that downregulation of ACE2 resulting from SARS-CoV infections contributes to disease severity by perturbing the renin-angiotensin aldosterone system, the same as presumed for SARS-CoV-2, given SARS-CoV and SARS-CoV doing the same manner. Okay. When you say downregulation, does that just mean that the cells statistically are expressing less of this thing? Like if you take an ACE inhibitor, is that just decreasing the percentage of expression? Okay. This is such a waste of time. I can't believe we were doing this. Why are you linking me this? Up, down, down. The structural biology of the SARS-CoV-2 spike protein and how it cheats the immune system. Okay. Hold on. Let's just finish this. Interaction between the S protein and the receptor. The interface between the RBD of the S protein and ACE2 is formed primarily by a gently concave outer surface of the extended RBM and the N-terminal helix of the receptor. There are 20 residues of ACE2 and 17 residues of RBD forming networks of hydrophilic side chain interactions. We don't care about that. Okay. Is this, how much is this mentioned? Do we care about this? Only one S protein mutation, D614G, exhibited clear evidence of positive selection, although acquisition of the furin site by SARS-CoV-2 and the resulting S protein cleavage appears to be essential for human infection. They also make the virus less infectious than SARS-CoV by rendering the S protein prone to S1 shedding, as discussed earlier. To compensate for this disadvantage, the SARS-CoV-2 S protein appears to have gained stronger intermolecular association between the S1 and S2 subunits by way of the D614G mutation. A number of studies support such a stabilizing effect as the mode of infectivity enhancement of the D614G variant virus. Cryo-EM studies on full-length dick sucking increase S protein stability. Okay, binding. A biochemical study clearly showed increased functional S protein density on this as a result of the reduced S1 shedding. Others have explained that a greater proportion of the 1-RBD up conformation among G614 containing S protein associated with increased virus infectivity. Most such studies, however, were performed with a non-native S protein lacking the Furin site. Holy fuck. Okay. Let's just go ahead and we'll just go ahead and add this bad boy right the fuck here. Fuck it. Okay. This study? No, stop! No more papers! Okay. We'll read this one and then we're done. Because this is a fucking waste of time, okay? Holy fuck. Spike proteins protrude from the SARS-CoV-2 viral envelope and are responsible for initiating fusion into human epithelial cells by binding angiotensin-converting enzyme 2 receptors on the host cell surface. That's our ACE2 meme. Due to their exposed location on the outside of the virion and their key role in infection, SARS-CoV-2 spike proteins are an important target for vaccine development and drug design. Over the last 2 years, many spike protein structures have been experimentally determined, providing essential details into the complex structural rearrangement that occurs after receptor binding and during fusion of the virion with the host cell, as well as into the interactions of spike protein molecules with antibodies. SARS-CoV-2 variants, particularly those associated with reduced vaccine efficiency, are strongly associated with mutations in 2 domains of the SARS-CoV-2 spike protein, namely the receptor binding domain and the N-terminal domain, which have both been structurally characterized. This review provides a comprehensive overview of the structural knowledge acquired over the past 4 years on the SARS-CoV-2 spike protein and its critical role in viral infection. We're not doing this whole fucking paper. Fuck you. What is the part that tells us about the up, down, up, down, up, down? Wait, is this? Okay, this is called that. Section 3.2 is the most relevant. The central stalk of the spike protein, responsible for viral fusion and entry, is formed of 7 domains. Listen to table 1. The helical core of the S2 subunit is formed by 3-stranded coiled. Wait, why would this be the most relevant? Don't we want to know about the up, down bullshit from the S1 guy? Why are you saying this is the most relevant? Okay, subunit 1. The S1 subunit, that's 2.2. I know. Just making sure you guys are paying attention, and you are. Good job. Clappa for you guys. Clappa for you guys. You did it. I'm proud of you. The receptor binding domains of the spike, which recognize and bind the ACE2 receptor located on human cells, are located at the top of each spike in the S1 subunit. Each RBD, 1 per promoter, can adopt 2 conformations such that it is oriented in the up or down position. When the domain adopts the up conformation, the receptor binding motif is exposed and is available to interact with a surface cell receptor ACE2 or antibodies. Not good. In its down conformation, the RBD interacts with neither ACE2 nor antibodies. The dynamics of this domain are thus relevant for immune evasion. An analysis of atomic B factors supports flexibility or instability of the down state of the RBD through increased disorder of the RBD compared to surrounding domains. Atomic B, what the fuck does that mean? No, don't care. Nope. Structures of the receptor binding domain and complex with the human angiotensin converting enzyme 2 ACE2 receptor reveal the interactions between the spike protein receptor binding motif and one of 2 binding site lobes of the ACE2 N-terminal peptidase domain. The contact between the receptor binding domain with an interaction surface area of 864 something squared and ACE2 with an interaction surface area of 820 is established at the bottom side of the ACE2 small lobe with the concave outer surface generating a total buried surface area. Even though the spike protein structure share up to 76% sequence identity, the receptor binding motifs share only 47.8% sequence identity. Mutation studies performed to evaluate how the sequence difference impact binding affinity to human ACE2 revealed that the substitution of amino acid Pro 499 I don't know what all these are. These are all amino acids, right? No! Nucleotides. These are all nucleotides. Right? And SARS-CoV-2 enhances ACE2 binding compared to SARS-CoV-1. Substitution of this amino acids to glutamine leads to substantial structural changes, removing an internal salt bridge between ACE2 residues, good one, that is buried in a hydrophobic, more like homophobic environment, and replacing it by 2 hydrogen bonds between each. Even though the spike proteins are SARS-CoV-2, okay, wait. After binding, there's arrangements, one domain that's spliced in fusion, directional region, which is the structure, with its RBD and down conformation, shows the EpiBR in an ordered state, packed around an internal. Okay, this doesn't. We're not going to find the answer to this, and it's not going to be relevant, and we don't care. Oh, wait, shit. Wait, that one did say that the furin cleavage happens in the cell. The spike protein is synthesized. The spike protein is synthesized on the rough endoplasmic reticulum before being transported to the Golgi complex. The targeting to the rough endoplasmic reticulum is done by the N-terminal signal peptide, which consists of 13 amino acids with helix-forming, highly hydrophobic residues. Does residue here also mean amino acids? Co-translational and post-translational processing result in the removal. Translation always refers to processing by a ribosome. Is that what that means when you say translation inside of a, is when RNA is being read? Okay. Co-translational and post-translational processing. Co-translational processing, I'm guessing, is like when the thing is being, when it cleaves itself as it's being read or something, I'm guessing. This sounds right. Okay. Co-translational post-translational processing results in the removal of this signal peptide in the endoplasmic reticulum lumen, trimerization of the spike protein, extensive glycosylation, and subunit glycosylation, and subunit cleavage by furin or furin-like proteases. Both subdomains are beta structure, rich segments located after the RBD. The N-terminal end of S2 is adjacent to a cleavage site targeted by furin or another protease during pre-processing of the spike protein. Holy shit. The host protease, furin, or some similar enzyme, cleaves the spike protomer at the S1-S2 interface before the virus gets secreted via lysosomes into the extracellular surroundings, and O-glycosylation has been shown to have both positive and negative impacts on proteolytic cleavage events. Therefore, differences in O-glycan expression may potentially modulate this protease activity and thus hurt infectivity. Okay. Okay. All right. We're good. Okay. We're just going to finish watching this and then we're done with all this.
Releases the first NSP, which is able to grab onto a ribosome and occupy it in such a way that the ribosome can only read viral RNA and not the host cell's own mRNA. This means that the virus takes over the cell's translation machinery, turning the host cell into a virus-building factory. Meanwhile, NSP3 cuts other neighbours before it cuts and frees itself. Eventually, the production of NSP stops because there's a pseudoknot in the viral RNA. This prevents the remaining RNA, which codes for proteins involved in viral genome replication, from passing through the ribosome. By now, a second cutting NSP has been made, which cuts itself and its neighbours. The translation can either stop here, or the pseudoknot in the viral RNA can slip, allowing the remaining viral RNA to be read. The second cutting NSP is now able to cut all of the remaining proteins.
Look at him go!
Because of the pseudoknot, the translation of the viral RNA often stops prematurely. As a result, the host cell produces fewer of the RNA processing NSPs from beyond the knot than the various membrane-modifying NSPs from before the pseudoknot.
Is there just like nutrients that are like floating around in, like, where do they get the energy for these reactions? I guess like the cytoplasm just like has like shit that you can eat where you're hanging out here. Yes, a lot. Okay. Cringe.
Brand is
Okay. Cool. All right.
Into species, it jumped into civets several times, and from civets to humans several times, and this is not what we observed with SARS-2. In fact, we see just a single jump and pretty low variability, genetic variability, so it seemed that...
10-question quiz time. Chat GPT. Can you create a 10-question quiz, a 10-question multiple choice quiz concerning the properties of the SARS-CoV-2 virus that an experienced political commentator, or how about that, an experienced public health official should be able to answer? Do not present the questions one at a time and hide each answer until I guess the answer. Which of the following best describes the genome of the... Oh, fuck, we got the retard version. Oh, no, wait, we didn't. I don't know what this means. Positive sense single-stranded RNA versus negative sense single-stranded... I don't know what the difference between those two are. It's one or the other. Good luck. What's the difference between positive and negative sense? Which structural protein of SARS-CoV-2 is primarily responsible for binding to the ACE2 receptor? We know this. It's the C1. Which of the following best describes the mode of primary transmission for SARS-CoV-2? Bloodborne, fecal, vector-borne, respiratory droplets, because we know that. Which of these is the approximate size of the virion? Fuck, I don't fucking remember. 20 to 4. Trying to remember that slide that we saw. Is it 20 to 40 nanometers? Transistor sizes, carbon atoms are... I'm trying to think of the length of shit that I know, that I've heard before. I don't know. A is my guess? 60 to 140, whatever. Which of these non-structural proteins is a key target for SARS-CoV-2 replication inhibition by antiretroviral drugs like remdesivir? Oh, fuck, I don't remember the mechanism. Non-structural proteins. RNA-dependent RNA polymerase membrane protein. Protease? RNA-dependent RNA polymerase? I don't know how remdesivir works. I knew that, obviously. Which of the following best describes the main function of the nucleocapsid protein? It protects and packages the viral RNA genome. Which of these best describes the SARS-CoV-2 classification within the coronavirus family? Alpha, beta, gamma, or delta? Is it a beta coronavirus? I don't remember. Who talks about this? Okay. Which of the following best explains why SARS-CoV-2 has a relatively high mutation rate compared to DNA viruses? It lacks any proofreading mechanisms. It's RNA-dependent RNA polymerase. There's no error correction at all. Are these the same thing? RNA viruses generally have higher mutation rates because their polymerase lack the same fidelity as DNA polymerases. Its replication occurs in the host nucleus, which increases mutations. That's definitely not true. I feel like these three are all saying the same thing, kind of. It's RNA-dependent RNA polymerase, no error correction at all. It lacks any proofreading mechanisms. I mean, it doesn't have... How can they proofread, no? Does that thing inside the cell count? It's RNA-dependent RNA. There's no error correction. Same fidelity as... I'm going to say A. Fuck! I don't know what polymerase is. It does have some proofreading capability via exonucleus NSP14. How could I forget that? But all right, well, overall, it's still more error-prone than DNA polymerases, explaining the high mutation rate. Which of these best describes the receptor binding domain within the spike protein? It's located in the S1 subunit. Which of the following best describes the function of the envelope protein? Binds to the viral RNA genome, functions as the main structural scaffolding of the virus. I think it's that one. What? Functions as an ion channel. What was it? Whatever. 7 out of 10. That counts. That's what we get on all of these, okay? Okay. The structure is not protein, it's a lipid.
SARS-2 was either pre-selected for human transmission or even pre-adapted for human transmission, which is, of course, much better explained by a lab leak than by zoonosis. And because under zoonosis, we still don't know how exactly it would have happened.
Okay.
And so basically in this section, I'll show that genetic data from early SARS-2 cases, they show a very low level of genetic variability, which is unusual for zoonosis.
Okay.
And also I'll show that the market was dominated by a later strain of the virus rather than the earlier strain, the earlier lineage. And like if we're to believe that the virus spilled over in the market, this is surprising because we'd expect the earlier lineage to dominate the market, and yet that's not what we observed.
Okay.
And so in totality, these observations, I think they're more compatible with lab leak than zoonosis.
Okay.
Okay, so in terms of the low genetic variability of the early cases, let me just talk about these a little bit. So basically, this is a diagram of 549 of the earliest SARS-2 genomes sampled between December 2019 and January 2020, basically the very start of the outbreak. And this diagram shows that the early cases split into two distinct lineages, each kind of radiating from this intermediate genome, AB genome. But what's striking is a small number of mutations that separate the root of the lineage from like the outliers. Basically, it's just like either 4 or 5 mutations, and I think that's a very narrow genetic spread, especially considering the number of viruses sampled here. And so such low genetic variability to me indicates that SARS-2 was already like pre-selected or pre-adapted when it encountered humans.
I don't like statements like I just don't know. I have no idea, and I don't think he's putting forth an argument to allow me to know if that's true or not. Right? Like I feel like a comparison with other viruses and showing their genetic variability on first crossover, spillover, but I feel like you need something more. I don't know. It's just low. It's just high. I have no basis of comparison for this, and I don't think 99.99% of the audience has a basis of comparison for this.
Also, the prevailing consensus is that lineage B of the virus evolved from lineage A because lineage B is 2 mutations farther away from the bat ancestor. But there's one research group that actually tries to claim that they were 2 separate spillover events. And this research group, led by Picard, proposed that the 2 lineages actually represent 2 separate spillovers from animals. And they base their claim on 2 main points. First of all, they just disregard any A and B intermediates, and like on this slide shows there's some A, B intermediates, but for their model, they disregarded them. They're saying like these are all sequencing errors. And second, they developed this Bayesian model to simulate viral evolution based on like sampling dates rather than just phylogeny, because phylogeny is pretty clear that B descended from A, but so there's like no reason to suspect 2 spillovers. But of course, with their model, like initially they had pretty considerable strong base factors, but it turns out that they actually had bugs in their code. And after you fix those bugs, that reduces the base factors by like 6 times and pretty much puts them into like very, very weak probabilistic support. Essentially, I think pretty much dismisses the hypothesis right there. But also, the genetic difference between those 2 lineages is just very small, just 2 mutations. And such a slight difference, I think that alone is unusual for 2 spillovers. And also these like 2 mutations can happen between, even in a single spillover, between like 2 consecutive transmissions. And here we have data from the Diamond Princess, the cruise ship, that show that in some cases you can have like 2 mutations or even 3 mutations in a single transmission event. So between 2 people, basically one person can evolve 2 or 3 null mutations. And I think this point is critical because it shows that the absence of intermediate genomes is not sufficient to conclude that there were separate introductions. So it's entirely possible that lineage E could have evolved from lineage A through a single transmission.
You already go back 15 seconds, you dropped out again.
Bro, he's not dropping out for us.
You cut out when you were saying that there could be 2 or even 3 mutations in something.
Okay, I was basically showing that in a single transmission between 2 individuals, you can have 2 or even 3 mutations arise in a single person. So the absence of intermediate genomes cannot be used as evidence that there were multiple introductions. It's entirely possible that with a single introduction, you can have someone evolving 2 mutations, like the difference between lineage A and lineage B. And I mean, I think Picard are the only ones who are positing 2 introductions, and there are several other models. None of them have 2 introductions. And moreover, as we've shown in our paper, Picard actually threw out many intermediate genomes that we think are valid intermediate genomes that should not have been thrown out. And moreover, after they published the paper, we found several more intermediate genomes from Wuhan itself that weren't included in the Picard model, and I think should have included. But I think that's all of this is a bit of a moot point, because after the fixing of the bugs, the errata that they actually now published, Picard has published, which reduced their base factors by like 6 times. I think by now this 2 spillover hypothesis is essentially like dead in the water. And also the earliest sampled lineage A genome already had an extra mutation away from the bad consensus. And this suggests that lineage A was circulating for some time before it was sampled. And so the timeline used by Picard to infer infection dates, because as I said, they use sampling dates as a proxy for infection dates. And I think it needs to be pushed back because probably lineage A was in circulation before, like for some time before it was sampled. And I think in general, why do they even care? Like, was it 2 spillovers? Was it 1 spillover? And I think they care because they're actually trying to explain why is it that the Huanan seafood market is dominated by lineage B, which is phylogenetically a later strain and obviously doesn't fit into the zoonotic hypothesis, because under the zoonosis, if B is descended from lineage A would have spilled over first, or the market should have been dominated by lineage A, as that's an earlier genome. But despite the phylogeny, Picard and others like Warby, they're trying to argue that it was lineage B that spilled over first in the market, but then lineage A spilled over second in the market, like a week later. And of course, but even under that scenario, you'd expect to see at least some lineage A in the market, whereas right now the market is almost exclusively lineage B, except for one sample for which I'll
I mean, I feel like it's pretty obvious that lineage B came second. That's why it's B and not A. QED.
talk about in a few slides. And so basically, to make that claim of two spillovers work, Picard et al. They have to rely more than just on phylogeny. As I said, phylogeny, B is clearly descended from A, so they have to use sampling dates as proxy for infection dates, and then use those dates to kind of infer when we see a certain lineage introduced into human population. But of course, I mean, that approach is in general is potentially flawed because early data is very scarce and it's anecdotal. When exactly did you have symptom onset? Some people could be mistaken, and especially here, like if we have just two people, one with lineage B and one with lineage A, one is saying, okay, I had symptom onset on December 10th, and the other side symptom onset on.
Does this guy ever present any salient arguments that support the lab leak theory? Well, this debate for this section is very much a debate on the genetic structure of the virus and then stuff related to like genetic evolution of the virus. So this is just going to be, all of this is going to basically be probabilistic. Now, depending upon what you're talking about, these probabilities can be very, 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 very high or very, 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 very low. But they're trying to argue probabilistically based on the evolution of the virus, like what is the more likely origination point, some human designed thing or some zoonotic transfer, basically.
On December 15th, and as I said, there's an extra mutation for the lineage A person, that could push back the date of the infection earlier. And also, as the market, it was noticed.
You're never going to have, unfortunately, even in viruses that we know to be zoonotic spillovers, it's very rare that you'll get the very first, like patient zero, and know the exact event. And then to say for certainty that it came from the wet market zoonotic origin spillover event, we'll probably never know the answer to that. But to say for certain that it came from a lab, you also probably won't know that unless the lab itself is cataloging and publishing information about the work they're doing, right?
The infection earlier. And also, as the market, it was noticed first, like the market was the biggest super spreading location that was noticed in Wuhan, and everyone with a connection to the market was sampled first because you actually needed a link to the market to get a COVID diagnosis in Wuhan early on. And so
that's not true.
We see that the market is almost exclusively lineage B, so lineage B had
I don't know if that came from the same paper that one guy sourced before. That was an incorrect reading of that paper. Maybe there's some other information that demonstrates that I'm not aware of that might get introduced later, though. Somebody said, why is that more likely than two spillover events? I think he was trying to argue, it sounds like he's arguing with reference to, or it feels like he's been arguing in reference to a zoonotic spillover theory that involves this coming from some bat-evolved coronavirus. And he's saying that lineage B is already multiple evolutions away from the lineage A strand. However, if lineage A came first, we would expect to see more people diagnosed with the lineage A1 versus the lineage B1. But they're trying to say that the reason why this happened is because there was just two separate spillover events or something. But he's saying that just doesn't make any sense. Lineage B was probably first. Yeah, something along those lines.
Potentially sampling bias, like it's been oversampled in the early cases. Therefore, whatever lineage A genomes, they had a disadvantage of sampling bias against them because lineage A was not present in the market and had no patients with a link to the market. And so this bias, it can actually skew the data when inferring spillover date from the sampling date. And that, again, I think I'm beating a dead horse in the Picard model, but by now I think nobody actually thinks there's been two spillovers. Sorry?
30 minutes.
45?
30 minutes.
Oh, 30 minutes. Okay. Right. Basically, in conclusion, I think it's very unlikely that there's been two spillover events, which I think by now it's pretty clear. Well, that said, even if there were two spillovers, I think it actually would support the lab leak hypothesis better, especially because it's just two mutations away between the two lineages. And we have examples of lab leaks multiple lab leaks from the same lab, like examples of SARS-1, for example. So it's entirely conceivable for a virus to leak from a lab more than once. But if there really were two zoonotic jumps, we'd expect to find many animals carrying the virus, because for two spillovers to occur like in the same location within a week and not anywhere else, I mean, that's really suspicious in itself. Why would there be this kind of exclusivity between the wildlife vendor and this particular market, and only that wildlife vendor would have these infect animals? And anyways, I think especially in light of how SARS-1 happened with like multiple animal spillovers. We had like 5 civet strains of SARS-1 and multiple human introductions, and we have nothing like this with SARS-2. And so it's just very surprising. And like this is 20 years later, we have like much more powerful sequencing technology, and yet just zero evidence of infected animals, infected intermediate hosts. Okay, speaking of SARS-1, when we look back at SARS-1, its emergence, like its pattern, is I think it's like very different from what we observe in SARS-2, because with SARS-1 there's been these like multiple spillovers over several months. Like the virus would get into human population, like try to get a foothold and go extinct, and then but eventually like it actually managed to start spreading human to human. But this is not what we observed with SARS-2. And also, it's not just SARS-1 that's had multiple introductions. Other viruses do the same. Like HIV had 4 different spillovers. MERS had like 50, by some estimates, 50 zoonotic introductions, and yet SARS-1, SARS-2 only had 1. So yeah, basically in summary, if we compare SARS-2 to other outbreaks,
so the guy before wasn't even getting. I think it was one of the judges asked, but this would have been my question: that if it was a spillover, 2 just seemed intuitively to me feels like a very unintuitive amount of spillover. Like I would expect there would either be 1 or like 100. That would be my just like intuitive assumption. Now maybe there might be a good reason why it could be 2 or why not, but well, I guess that'll be in his counter argument.
SARS-1 stands out as a clear, SARS-2 stands out as a clear outlier, because normally a virus would jump multiple times from animals to humans and infect them usually at different locations, because there's some sort of reservoir of this virus present, and there's no reason why it wouldn't be able to infect other members of other species at multiple times. But with SARS-2, we have yet to find a single infected animal. And so I think the evidence points to a concentrated human spillover event. And additionally, the A and B lineages of SARS-2 are only separated by 2 mutations, which is unusual for zoonotic spillovers, because typically you see multiple strains with multiple differences between them. Okay, let's now focus on the Huanan seafood market, because I think the entire zoonosis narrative is based on the Huanan seafood market being the location of the animal spillover of the virus. And I think it actually, there's data that, like the observations we have is more consistent with the Huanan seafood market being just a super spreading or amplifying location of lineage B. Because, first and foremost, lineage B is a later strain, and market is almost exclusively lineage B. And this is inconsistent with zoonosis because, usually the earliest strain should be dominating the spillover location, like the market. And yet the genomic data of the earliest cases we shows that it was exclusively lineage B in the patients. Like all 16 of the earliest Wuhan patients linked to the seafood market, all of them had lineage B. And this is surprising because lineage A it had 2 mutations closer to that ancestor, but you'd expect, if that was the spillover virus, you'd expect at least some people from the market to have that lineage. But also additionally, the environmental samples also all but 1 were lineage B. And the only lineage A detected in the market from environmental samples wasn't even from the environmental sample. It was from a cultured sample based on an environmental sample. But that environmental sample only had like 22 SARS-2 reads and had a very high CT PCR value, and yet they somehow managed to culture a virus, like a viable virus from that sample. But that virus, when they sampled it, not only had lineage A mutations, it also had like 2 or 3 additional mutations that weren't seen until much later in the outbreak. They were like, yeah. So basically all of this is more in line with the contamination, that sample being a result of contamination either during culturing or sequencing, and but not like a bona fide virus from, bona fide lineage B virus from the market. And so yeah, basically because of this kind of overwhelming presence of B, like almost exclusive presence of B in the market, I think it's much more in line with the market being just a super spreading location of lineage B or amplifying location of lineage B that got into the market after lineage A spilled over in Wuhan earlier and basically developed into lineage B, and then lineage B got into the market.
I don't remember for zoonotic spillover. I don't know if it's required, if it has to be that there were 2 spillover events, or if they have some other way of explaining it. I don't know.
Amplified. And the early kind of interplay between lineage A and lineage B in Wuhan is interesting, because even like in the early stages, at least a third of cases in Wuhan were lineage A. And yet, we see no lineage A in the market, which is strange. And of course, lineage B had a transmissibility advantage and ultimately outcompeted lineage B into extinction. And so in this case, in this diagram, M is same as lineage B. It's just an old diagram. And all of this is not consistent with zoonosis, but I think it fits very well into a elablic scenario. And even more, some of the earliest genomes found later during sampling were even closer to the bat ancestor than lineage A. So they had like either at least one more mutation closer to the bat ancestor than lineage A. And so these findings suggest that there might have been an earlier strain that spilled over, like even earlier than lineage A, which then, eventually mutated into lineage B. And one such mutation that's closer to the bat ancestor is this mutation here, C18060T. And several genomes have been found that have this mutation, several early genomes in circulating genomes, and also in some like forensic metagenomic analysis. For example, here this Arctic soil data set that was sampled and sequenced, sampled in Antarctic, but sequenced in Wuhan in like early 2020. They found these SARS-2 genomes from potentially early patients that have this ancestral mutation. And Jesse Bloom analyzed these genomes and reported that there were numerous reads consistent with these being progenitor genomes, suggesting like an earlier SARS-2 in circulation in Wuhan. And of course, Jesse Bloom also is kind of famous for finding these deleted patient data set that had like 11 patients, I think 4 of which had even earlier genomes. Three had one mutation earlier, and one had maybe even 2 mutations earlier, like closer to the ancestral strain than lineage A. And the exact number of mutations is not really important. I think the significant finding is that lineage B was already was not the ancestral lineage, and yet the market is dominated by lineage B, and this is inconsistent with zoonosis, because we would expect if the market was the location of zoonosis, of spillover, you'd see at least some lineage A in the market, if not like be dominated by lineage A. But, and as I promised, there's only one lineage A sample in the market, and out of like, I think they had 25 or so positive samples, environmental samples. They had 16 patients, all of them lineage B, and then they had 25 environmental samples, and there's only one lineage A from a cultured sample. So the original, and I already mentioned this, the original glove sample had only like 22 SARS-2 reads and had a very high CT value for the PCR screening, but yet they managed to isolate a viable virus. And so with additional mutations, like I said, additional mutations not seen in Wuhan at the time until like months later. So all of this is really more consistent with contamination during sampling or sequencing. And this keeps... So yeah, basically, I think I also... made this point several times, is essentially like the dominance of lineage B in the market is just inconsistent with the market being a spillover of zoonotic lineage. And unlike SARS-1, which had multiple spillovers in multiple locations, multiple animals, with SARS-2 we see none of that. And so this is very surprising, and I think more in line with lab leak than with zoonosis. Okay, mutation rate. So basically, I think the virus exhibits a very low mutation rate. Like even the early virus exhibits a low mutation rate, in contrast to like SARS-1 or other zoonotic viruses. And this is surprising because you'd expect a virus that spills over to a new host to have a very high mutation rate, especially initially as it adapts to this new host. And here's some diagrams of these like low mutation rate, and I think basically, yeah, they show that from the start it's pretty consistent at like 20 mutations per year. And I think Peter actually challenged this claim and had a figure of 37 mutations per year for early genomes. And so I looked at the paper he cited in this estimate, and it was interesting because he said at the same time that it had 36 mutations in deer, so basically fewer mutations than in humans, but the paper actually had much higher mutation rate in deer, like 1.6 versus 1.3. So if you actually convert it to the full genome at like 30,000 nucleotides, I think you arrive, this should be like 48 to like 52 or something, so not 36. But it's a good paper. I like it because it shows basically compared to alpha and compared to delta, the early mutations in the early strains, and sorry, this is the red is the early strains, the pink is the later strains, and blue is deer. And basically you see deer is still higher. Like in their estimate, deer still has higher mutation than the earliest genomes. And the earliest genomes used by that paper come actually from Picard et al. Picard is everywhere. And Picard actually, their original data, based on which the Deer paper used for their analysis, they estimated 27 substitutions, 27 mutations per year based on these slopes rather than 37. So, I mean, again, it's. I think the exact number is less critical. What's critical is that even if, I mean, yeah, clearly there's potentially like the first few months of the outbreak, you have a higher mutation rate. But of course, that data is also noisy because Picard all they use from like December to February. And the earliest sampling dates, as I said, are very sparse. And
Fuck, I feel like for a debate like this, actually, now I changed my mind. I don't think this is a good structure. If both people are pretty educated, I feel like this is far better dealt with in an academic paper writing setting. Because this guy is saying so much, and for this guy's going to give a response, which will be nice, but it's going to be very hard mentally to keep track of every claim that this guy's made, although he'll try to address them. This would be done much better in a back and forth manner, writing papers. Or, nobody likes to do this. It would be really interesting to hear these guys go through a debate like this over like 4 or 5 hours, like claim by claim by claim, resolving them. Just because, like I said, I can't evaluate anything here. And he's like, oh, this is likely to happen. This is unlikely to happen. I'm like, I don't know. I truly don't know. I don't have any basis by which to have an intuition there. But I guess we'll see how we'll see how his response is.
Essentially unreliable because they're anecdotal. But even if you rely on them, the difference between the mutation rate between like early strains and later strains is like twofold between early and later. So basically, they had like 27 versus 20.
Yes. Now do you understand why we do it via papers? No, I never ever knew that at all. I always thought that writing papers, I thought all academic papers were stupid and a waste of time. But thank you. Now I finally, on July 5th, 2025.
20, like if you take per car, or 27, like
15 minutes.
Okay. Right. 15? 15 minutes? Okay, I'm good. Right, so basically, where was I? Right, so the Picard estimate.
How do you tell when someone is trying to bulldoze you with stats/academics as opposed to logic/substance? Well, that would happen in a live back and forth. You can't really bulldoze in timed formats like this. Even if somebody puts a bunch of bad arguments, it's not really bulldozing. They both have 90 minutes to get their point out.
For the early mutation is, for the earliest strains, the mutation rate is 27. The later, like next strain number for mutations is 20, like the lowest is 18 per year. And the Deer paper estimated, like it had not the total mutations, but substitutions per site per year, it had like 1.3 for the early genomes and 0.6 for the later genomes. So basically, at most, it's like a twofold difference between the earliest and the later stages of SARS-2 evolution. And at the same time, when you look at what happens when SARS-2 spills over to different animals like deer or minks, its mutation rate gets increased much higher. So for example, here it's like between 4 and 13 times higher mutation rate when it's spilled over into minks. And yeah, basically, I think a low mutation rate. I mean, under the lab leak hypothesis, it's very easily explainable that the virus was already kind of pre-selected for human transmission, but and you know maybe even further adapted. At the same time, I mean, yeah, it could happen naturally as well. Like all of the features that we see in SARS-2 potentially could arise naturally and then spill over naturally. But I think it's like in theory at least. But like the number of features that the virus would need to arise, you know, at the same time or very nearly at the same time, I think it's very unlikely for them to arise in a single animal or like single species, especially like definitely not in bats, because as I mentioned. The Furin cleavage site or the absence of this aniline glycan, both of them are detrimental to transmission in bats. So it's very...
Cryptic transmission means basically an asymptomatic transmission from one thing to the next, and it's like undetected, right? Because it doesn't present with enough symptoms or whatever to get measured in a lab or treated.
It's surprising for these features to have arisen in SARS-2. But without them, of course, SARS-2 would not be suitable for human transmission, would not be a respiratory virus, would not infect humans. So even despite it having this kind of high affinity to the human ACE2 receptor, without a Furin cleavage site, it's still an enteric virus, and without this ablation of this glycan, it would be like 60 times less infectious. So it's just, yeah, the number of things that would need to happen at the same time in like between the bat virus and the human transmissible virus are just, to me, a very, how should I put it? Well, surprising, unexpected. And of course, for all of this to emerge naturally, this would probably need to happen close to Wuhan, like emerge naturally in Wuhan, because of course the natural habitat for these viruses and for these bats is like 1,000 miles away from Wuhan, like in southeast Yunnan or like Laos or Vietnam or whatever.
Are you wrong about the FCS without telling us? I have no idea. I know less about the Furin cleavage stuff than I do before. Something it cleaves something. I don't even know what cleavage means now, but it cleaves something. My understanding is that when the spike protein connects to the cell, when the RBD binds with the ACE2 thing, somehow Furin cleaves off the spike, the S1 subunit, and then the S2 injects it, and they fuck each other, and they all go in. But the guy in chat, the MIW, he says that the Furin does not cut off the S1, the subunit of the spike protein, and instead, when the virus is being manufactured inside the cell, that Furin cleaves it when it's being produced. I don't know what the correct answer is. I don't know if it matters that much. I think the MIW in chat is saying that the Furin, when it cleaves the spike proteins, is what's resulting in the up configurations for the RBDs. Or no, the receptor binding motifs, the RBMs. In the video, it said that I think in the video, did it say Furin cuts off? What did she do? I don't know.
Making contact with the host cell and starting the infection. It's glycosylated, meaning it has a coat of sugars. When the virus closes in on a host cell, its spike proteins open up and bind to a receptor on the host cell called ACE2. After the spike protein makes contact, Furin cuts off the outer part of the spike protein, called the S1 domain.
So he's saying that that's wrong. Furin doesn't cut anything off here, but rather Furin does something inside the cell while it's being created. Nope, I didn't say that. Okay, I have no idea what you're saying. I'm not sure. I don't know. Here, another picture. Okay. Okay, virus connects, internalization. Once it gets in here, when does the S1 part happen? Am I retarded? Am I just missing it? So it enters here, endosomal acidification. I don't even know what that means. That cleavage is caused by TMPRSS2, not Furin. This is cleavage of S2 though. Isn't that different than S1? Like S2 is when it binds to the S2. Isn't S2 what allows for the fusion? That's what's happening here. When the S2 thing gets cleaved, then it fuses to the cell membrane and then it injects into the cell here. No? Actually, wait. This is saying the S2 site is here. It sheds the S1. FP, is that Furin? That's Furin protease, isn't it? Wait, I think what you just showed me, is this not saying exactly what we said the whole time? So Furin protease is what comes here and cuts this is what happens with the shedding of the S1, the subunit 1 of the spike protein, and then this cathepsin or whatever is causing the fusion into the membrane. Oh no, it is fusion peptide. It's not it's not Furin protease? Conformation change inserts FP into target membrane. Oh, fusion peptide into target membrane? I get maybe, okay. I don't know. Why are there two FPs? I don't know. What you're saying is wrong. Stock 20. What is he saying? Destiny, do you know what TMPRSS2 protease cleavage site is? I thought TMPRSS2 was just another way to get into the cell. Like you could get in via ACE2 or you could get in via TMPRSS2. He thought the furin cleaved at viral entry for the FCS. They were talking about the FCS that they were talking about was cut in the cell, Golgi during viral maturation. The cleavage is caused by TMPRSS2, not furin. I thought this was just another gate into the cell. Does this cleave shit? Does it cut shit up? This gene encodes a protein that belongs to the serine protease family. Encoded protein is a type 2 transmembrane domain, a receptor class A domain, a scavenger receptor, a protease domain, some protein. This is the domain of this protein. The protease domain of this protein is thought to be cleaved and secreted into cell media after autocleavage. This protein also facilitates entry of viruses into host cells by proteolytically cleaving and activating viral envelope glycoproteins. Viruses found to use this protein for cell entry include influenza virus and the human coronaviruses HCoV, and then SARS-CoV-2 is here. This protein also facilitates entry into viruses. I thought, isn't this similar to like it can get in via this, or it could get in via like ACE2? National Library of Medicine, hold on. Bro, I don't think I need to know this. I don't think any of these are ever gonna get. I think you can read the commentary that accompanies that figure. We will never need to know it at this level of depth, okay? Okay, 2 spike protein cleavage events are typically necessary for the coronavirus entry process. One at the junction of the S1 and S2 subunits, and the other at the S2 prime, is that what that thing's site? Internal to the S2 subunit. In the case of SARS-CoV-2, the polybasic sequence at the S1, S2 boundary is cleaved during virus maturation in an infected cell. So this is when it's being produced in some part of the Golgi or the endoplasmic reticulum. Some part is cleaved during the virus maturation in an infected cell. But the S2 prime site is cleaved at the target cell following ACE2 binding. Virus binding to ACE2, step 1, induces conformational changes in the S1 subunit and exposes the S2 prime cleavage site in the S2 subunit. Depending on the entry route taken by SARS-CoV-2, the S2 prime site, this is prime, I'm reading this right, S2 prime site is cleaved by different proteases. Left. If the target cell expresses insufficient transmembrane protease, then serine 2, TMPRSS2, or if a virus ACE2 complex does not encounter TMPRSS2, the virus ACE2 complex is internalized via clathrin-mediated endocytosis into the endolysosomes, of course, where S2 prime cleavage is performed by cathepsins, which require an acidic environment for their activity. Okay, so on the left is, it depends on the entry route taken by SARS-CoV-2. So if there's no TMPRSS2, then it gets sucked in via some other process, and then the S2 prime cleavage is performed by cathepsins, but that requires an acidic environment for activity. Okay, and then in the presence of TMPRSS2, in the presence of S2 prime cleavage occurs at the cell surface. In both entry pathways, cleavage of the S2 prime site exposes the FP is fusion peptide, and the dissociation of S1 from S2 induces dramatic conformational changes in the S2 subunit, especially in heptad repeat 1, propelling the fusion peptide forward into the target membrane, initiating membrane fusion between viral and cellular membranes. It forms a fusion pore through which viral lineage moves into the host cell cytoplasm for uncoating replication. Several agents disrupt interaction between the S protein and ACE2 mimetics. Therapeutic monoclonal antibodies targeting the neutralizing epitopes on the S protein and antibodies. Contrast, strategies targeting post-receptor binding steps differ between the two pathways. Being a serine protease inhibitor, CMOstat restricts the TMPRSS2 mediated entry pathway. Hydroxychloroquine and chloroquine block endosomal acidification, which is necessary for cathepsin activity, and thus restrict the cathepsin mediated entry pathway. Jesus. Jesus Christ. Okay, listen, this is whatever you want it to be. I don't know. Tomorrow I'll be a refresher mind, and maybe I'll go back and look at this, okay? I have to finish at least this video. Either we're going to be done with both of these today.
And so it's just very odd for this to happen without leaving any trace, like in between. Like if it's wildlife smuggling routes, to see no evidence of this spilling over along the way or in some intermediate hosting locations of these wildlife, and yet not see any evidence of a single infected animal, like that's very, very, very surprising. Okay, so I think pretty much done. I mean, this is the final slide, the best explanation slide, and this is the kind of the root claim process where before assigning probability scores, the base factors, basically it's trying to steelman both hypotheses, presenting like the best arguments for each hypothesis, and then comparing evidence for both. And I'll just leave this slide to be explained by SAR in session 3, I guess. And this is like a preview of the results, and I think the lab leak probability here remains extremely high, of course, based on these best explanations. And I guess we have some time for questions, right? So we have like 8 minutes if I have my timer correctly. If you'd like to ask some questions, finally, I'm surprised that I actually finished before my time, or we can leave this time for later answering questions, or I'm not sure. But basically, at this point, thank you. If you have any questions, feel free to ask.
Thank you, Yuri. That was very interesting.
I'd like to go back to that kind of my initial question about
this guy has 99.9% probability. Sure, but remember, this whole like rationalization or this Bayes whatever bullshit is stupid, because it all comes down to the underlying assumptions, okay? This guy though, I don't know if these are correct or not, his underlying assumptions, because I don't have the background to know. I can't say. However, I will say that he did way more work in trying to justify where he was getting these modifications or how he was estimating these probabilities. He put in way more work to that. So even though the probability that he's correct here is very, very, very high, at the very least, he put in a lot of work into justifying these underlying things. He wasn't like, big cities, we multiplied by 2. We divided all of the population of all of Southeast Asia by the city of Wuhan to get 1.4. Like, he put a lot of work into figuring these. So, I'll grant that. At the end of the day, these calculations are whatever, but the underlying examination of the facts is the most important part, I think.
Kind of what I'm struggling with, or what I'm trying to evaluate, is how much should I think it is suspicious or coincidental that the diffuse proposal contains a whole bunch of things that look like genomic features that would be expected, or like that would produce genomic features that we then see in SARS-CoV-2.
That's a really good question, right? What are the chances that we spent so much time, what would the comparison even be? I don't know, that we're like researching how to make air conditioners cheaper for houses, and we're trying to figure out, like, can we, retrofit a bunch of apartments in Seattle and London with AC, because not everybody has it, and then all of a sudden we start to get heat waves, and it's like, holy fuck, did the liberals create heat waves when they were researching, how heat waves would impact? And it's like, well, climate change, right? It's a predictable thing. That's a very good question.
And so the spirit of my question was, one thing that would help me evaluate that is if there was any sort of data trail or any sort of kind of evidence that any of the diffuse proposal specific experiments were being carried out or were being used as a blueprint. So as an example, if you go to your slide 41, Yuri.
Yeah.
Okay, so you're citing this paper here. You cited the FAL1 restriction site is very interesting. And here in this paper, DNA amplicons were digested using FAL1 with CutSmart buffer purchased from New England Biolabs. Mm-hmm. So this is exactly my question. If this research is done, where, if any, paper trail exists, and if it doesn't exist, why haven't we seen it, given that U.S. vendors are frequently the suppliers of a lot of these things?
Right, well, I mean...
That's going to be way outside of the... It's a good question, but I don't know if he spent any part of his presentation addressing like the supply lines for, the labs. I'm curious what
given that U.S. vendors are frequently the suppliers of a lot of these things.
Right, well, I mean, restriction enzymes, they probably have a stock of, them and buying them up years in advance and just would use it. And of course, these days, most of the scientific supplies are available in China as well. And so any paper trail you would expect would be in China. And obviously, it wouldn't be released. I don't think they would have used any US or other vendors for their internal like WIV. And they, I think over the years, they also tried to in-house most of the capabilities, like both sequencing capabilities and synthesis capabilities. But even if they need to...
So that might be true, it might not be true. I wish he had a source to validate that. I'm willing to give a bit more leeway to him because he is kind of like, I think he's like a subject matter memer. Like people are linking China, he actually has like published stuff on this. The genetic structure of SARS-CoV-2 does not rule out a laboratory origin, and this is published in Bioessays. I don't know the strength of the journal or not, but still, regardless, he's got he's credentialed at least a bit in here. But it would still be nice to see something that he could point to externally showing, in-housing of WIV stuff or whatever. But again, this wasn't necessarily like the logistics weren't really part of his argument as much.
Capabilities and synthesis capabilities.
Are you studying this in preparation for something or just seeing knowledge? Well, one is general expansion of knowledge. Policies is just really boring to me right now. I hate it. It's gotten like so ridiculous. Like tomorrow it could be like Trump rapes like 7 kids and like Charlie Kirk is tweeting out, like Biden raped 8 kids, or those kids actually love Trump dick. And then like in 2 days we'll all forget it. It feels really goofy to me. And I like researching Israel Palestine, so we're just gonna spend the next a little while, I don't know, just researching stuff. Also COVID shit and COVID conspiracies and lab leak and all the conspiracy theories are right. Like it gets brought up over and over and over again. So I'd rather, it's an interesting thing for me to study and gain knowledge about.
But even if they need external help, I think they would have used Chinese vendors. There's no need for them to.
I don't think our future stuff will be as sciencey heavy though, or whatever. The next topic that we do, I might like read the Mueller report or something. It won't be as, or we'll do like Herd versus Depp. Yeah, it won't be like as, whatever this is.
And yeah, I mean, synthesizing DNA is these days outsourced to China. Anyways,
you like talking about whether left-wing populism is on the rise in America? I don't get right.
Most cheapest.
I mean, I think that's a debatable question. I don't that
synthesizing DNA is these days outsourced to China. Anyways, the most cheapest.
I mean, I think that's a debatable question. I don't that assertion seems without evidence. More broadly, I buy a bunch of nucleotides from the United States that are synthesized. I think my question is broader than that. Is there any evidence?
Okay, Chief.
Like I understand what you're saying, that there's a lot of Chinese vendors that can supply this. That makes total sense, and that there wouldn't necessarily be an accessible paper trail. Perhaps even if it was in the United States, this information could be deleted or it could be hard to come by. But so they're like, I'm just asking.
You are making this Furin cleavage site thing sound super complicated. It is not. The science is settled. When you do this, you are hurting your science communication and lowering the trust your community has for you. True. I did see that Reddit post yesterday where somebody posted Destiny's inability to accurately articulate the functionality of the Furin cleavage site inside of the SARS-CoV-2 virus is hurting my ability to trust him for... Furin, a kind of protein convertases, PCs, is located in the seriously, it's affecting sciences now. Golgi network and activated by acid pH. Furin can cleave precursor proteins with specific motifs to produce mature proteins with biological activity. The first and fourth amino acids in the N-terminus of the substrate cleavage site must be arginine, RXXR, any amino acid, demonstrated that the spike protein S1/S2 and S2' cleavage sites of the infectious bronchitis viruses beta strain can be recognized by furin, which is a distinctive feature of IBV. Based on the characteristics of furin's recognition substrate sequence, some short peptide inhibitors have been developed, such as decanol arg a modified alpha 1 intradonus. Okay. Through a series of analyses, this study proposes that one of the important reasons for the high infectivity of SARS-CoV-2 is a redundant furin cleavage site in its spike protein, and through structure-based virtual ligand screening and in vitro enzyme-based assay, the antiparasitic drug diminazine was found to show competitive inhibition on furin with IC50 of furin cut site and spike protein. Okay. By sequence alignment of spike protein sequence of SARS-CoV-2 with its highly homologous sequences, it was found that the spike cleavage site of SARS-CoV-2 possessed 4 redundant amino acids, PRRA, and these were not found in those of high homology sequences, thus forming a furin-like restriction site as RRAR. Through prediction and the sequence is easily digested by furin. To explore the evolution of the sequence, we use BLAST. What part of this am I supposed to read? Holy shit. Peter Miller offered to come on your stream. Okay, I'll do it after I watch all the debates. I have to watch all the debates first, okay? To further explore the possible mechanisms of furin cleaving SARS-CoV-2 spike, we perform protein-protein docking. Hell yeah! Woo yeah! Okay, thank you. Okay.
Asking if there's been, kind of any evidence at all of diffuse activity work that was carried out at WIV using this as a blueprint. Like, we have a bunch of speculation, which certainly seems plausible, but is there any other evidence that they did specific things as proposed in WIV? I mean, the one that stuck out to me is like that primer walking. It seems very unlikely that a graduate student, if they were going to primer walk to sequence these viruses.
What is this?
The way you feel. One kiss is all it takes. Falling in love with me.
Okay, true. I feel like cells and stuff are fun when the smallest thing you ever have to look at is a like an organelle. Past that, it's cringe and fake, and probably not even real, which is why it's fake and cringe.
They did specific things as proposed in WIV. I mean, the one that stuck out to me is like that primer walking. It seems very unlikely that a graduate student, if they were going to primer walk to sequence these viruses, would be doing that synthesis.
Jesus.
I mean, I'm not aware of any paper trail. I'm not sure if anybody looked at the paper trail outside of, like, I don't know, intelligence agencies. But yet, again, yeah, my expectation would be that they wouldn't be using, providers outside of China. Like, why? There's very, very strong capability in Chinese, like, biotech providers, and so.
Okay, that's helpful. Thank you.
While we're talking about diffuse, I did took some time to briefly look at the diffuse grant, and it sounded like a lot of this work was, as proposed by diffuse, would have been done in America, specifically in UNC. And the sorts of things that I would expect could lead to a development of a SARS-CoV-2-like virus would not have taken place in WIV as proposed in the grant. Can you account for how in the lab leak theory this virus would have come out of WIV instead of a different lab? Can you explain this discrepancy between the diffuse proposal and the lab leak theory?
Yeah, well, I think I mentioned in the beginning that, diffuse was not gospel. WIV could have done whatever they wanted for their own research. Diffuse is what, EcoHealth wanted to do and get money for EcoHealth. And while in the diffuse proposal, the Furin cleavage sites were supposed to be done by Ralph Barrick, of course, nobody is prohibiting WIV to later do this kind of work, especially because diffuse wasn't funded, and especially it seems that kind of Barrick and WIV parted ways on their Furin cleavage sites. But a lot of the work, even within diffuse, a lot of the work was supposed to be done by WIV, especially with live viruses. So while Ralph Barrick could have been, well, again, yeah, this could have been done, but under diffuse is really not a good exercise, because diffuse is not limiting WIV to what things they could be doing. They could be, if they have an interest in synthesizing, inserting novel Furin cleavage sites, they can do this locally very easily. They have this capability. And so just because diffuse was talking about Ralph Barrick doing it, I wouldn't give any weight to WIV not being able to do this or not wanting to do this.
Okay, what is your contention then? The MIW? So Furin acts at it can cut up here, or it can cleave up here, and it can cleave down here? Is that what you're saying? Because in this little graph, it's on both areas. So Furin is up here, cleaving up here, the Pac-Man guy is up here, and then down here it's in the endoplasmic reticulum Golgi. It's in both places? It's all over here. It's eating everything. Yep, but the other proteases cut it up there. Okay, so Tim is cutting it up a lot. So that one video was just wrong when it said that then, right? Are we saying that video was wrong? The British lady was incorrect when she said, no, Furin binds up there, but then it cleaves down there. I don't think I can understand this, my dude. I don't think I have the background or whatever to, because everything you're linking me it's not saying enough. What? Furthermore, Glock and Simmons et al. Have demonstrated that SARS-CoV spikes can be activated by cleavage in two ways. Oh, okay, when we say cleave, what the fuck does cleave even mean now? Does cleave mean cut? Or does cleave mean something different? It means cut, right? Okay, spikes can be activated by cleavage in two ways, including proteolytic activation by capsaicins B and L in host cells, where an inactive protein precursor is converted into its active form through the cleavage of one or more peptide bonds. Okay. In addition, SARS-CoV spike can be activated by TMPR SS2 cleavage on the host cell surface. When it says the spike can be activated, does that mean it can bind? Is that what activated means? Or does it mean activate as in once it's activated, it's cut off and then it's shed? As we can see in figure 2b, the spike protein of SARS-CoV-2 can be cleaved at multiple stages, which greatly increases the efficiency of fusion. Demonstrated that the CS1 of the S protein of SARS-CoV was easily cleaved in the host cell, and they mentioned the TMPR and the cleavage of CS2. It is likely that the virus will fuse with the cell in the cell plasma membrane and release the genome into the cells. In addition, the receptor affinity of the cleaved spike is also greatly enhanced. He wants you to click the show full caption under the figure you were just looking at. I think that was just a picture. I don't think I can click anything. Oh, this show full caption. A. This is the sequence analysis of spike proteins in SARS-CoV-2. It contains an N-terminal signal peptide, S1 and S2. S1 subunit 1 contains the N-terminal domain and receptor binding region. S2 is mainly responsible for membrane fusion. The C-terminal region of S2 is S2 prime, and it contains a fusion peptide, HR1, HR2, and a transmembrane domain. The amino acid sequence numbers of every domain are annotated below. Cleavage sites contained in SARS-CoV and SARS-CoV-2 are marked by rhombus. B, a schematic diagram of the process of SARS-CoV and SARS-CoV-2 infecting those cells. This is SARS-CoV and this is SARS-CoV-2. These proteases are presented by sector in different colors. Furin can cleave spike in the process of viral maturation. Is this what you want me to read? That it could cleave the spike protein in the process of viral maturation? Is that what you want me to read? Is that like desirable? Does that help? Or when it cleaves it, when it cuts it there, wouldn't that inhibit the ability of the virus to go out and do shit? Or does that just help it infect more shit inside the cell? Okay, listen. Okay, there, it does that then. There, I've said it, I read it.
Especially if they wanted to do their own stuff for their own funding, internal funding, and not be kind of beholden to NIH or EcoHealth or whatever.
But just to be clear, do you have a reason to say that Ralph Barrick stopped collaborating with people other than the fact that they weren't co-authors on a specific paper? Or I mean, obviously,
yeah.
Because I don't know the people involved, it's very hard for me to have a feeling for who is working with whom, why, that sort of stuff.
Right. No. Basically, the paper, not paper trail, but the paper is published by Barrick in like 2019 and afterwards. Especially this particular paper that is precisely about the things that they collaborated with WIV in the past several times, both diffuse proposals.
It pre-activates in the cell for higher binding affinity because it is mostly an RBD downstate, which is hidden. I don't understand that. It doesn't make sense that the virus would proliferate in the body with the up configuration, because it sounds like one of the whole advantages of the up and down configuration is that when it's in the down configuration, it is hidden from the body's immune system. But if it proliferates through the body in the up configuration, then it loses that whole advantage, and that sounds like some kind of advantage. It sounds like there's some way for it to configure itself up and down based upon how close it is to a cell or something like that. That's what it seems like. So when you say that, like, oh no, it gets turned up before it even leaves the cell, well then then it sounds like it doesn't help that much at all. Then the glycogen stuff for the glyco bullshit, binding bullshit things. Look up what disc on the Q said. Why does this part matter? I don't need to understand this. I don't think it's important to any of this. I'm looking at like 15 papers on this. I don't understand. My brain is too small. Fuck. Now I dropped a thing. I don't care. It's gone. Figure 7. What? Fig. Point 7. Nice picture. Fuck. The 4 trimer configurations observed in our data suggest a potential mechanism for receptor-induced triggering that involves sequential activation of protomers. In this model, we speculate that DPP4 binding to transiently exposed RBDs functions as a molecular ratchet that drives the trimer to the 3 RBD out open conformation. So every now and then a single RBD weakly pops up, and then this DPP4 binding thing happens, and that makes it all come out. This conformation is likely intrinsically unstable because the RBDs no longer help mediate trimerization of S1 or sterically inhibit refolding of S2 by sitting atop the central helix. In support of this model, we note that the 3 RBD out conformation is only observed in 0.3% of our cryo-EM data. Further experiments will be required to test this proposed model of receptor-induced triggering, but sequential activation of promoters in a class 1 fusion protein has recently been demonstrated by... What? I don't know why... It turns up and down constantly. RBD up-down exists in dynamic equilibrium. It's about balancing immune evasion with receptor binding. That doesn't make any sense. That sentence doesn't make any sense. The cell doesn't know where the immune... It's not like it drives. It's not like it turns on like rocket boosters and drives to cell. It just floats around in the fucking body. If it's up and down 50% of the time, half on or half off, then there's no advantage between just being always off or always on. It doesn't... The up and down doesn't matter if it's just random. At that point, it might as well just be on all the time or off all the time. Because it's not like it knows when it's close to... It's not like it knows when it's close to a cell unless there's some other mechanism at play. It is constantly dynamically switching up and down, but when it binds to ACE2, it stabilizes, right? If it's constantly switching up and down such that it can find ACE2, then that would mean that it's immunogenic and can be found by your immune system. Then there's no advantage to it ever being down if it has to be up to find the ACE2 receptor. There's no advantage. At that point, it might as well just always be in an ACE or in an up configuration or whatever, no? Yes, it does, because it only needs to be on when it's close. But how does it know when it's close? It doesn't have eyes. There's not a pilot. In the virion, there's not a pilot driving it to the cells. Why are you like? I don't know. I don't even know what these words mean. Cryo-EM structure of the 2019 nCoV spike in the pre-fusion conformation. Bro. Does this even talk about the ACE2 binding does not compete with immune discovery? Are you sure? Because it sounds like it looks for the receptor binding domain sounds like the part that is the most likely to be eaten by the immune system part. I thought we read that in a paper. That the way that the immune system would deactivate it is with competitive fucking immuno-suck-offness. If you can suck all the loads out of the binder and attach your mom's mouth to all the dicks, then it can't go rape any of your cells to make more viruses. Your immune system can't see anything either. It's just shit bouncing around at random, and sometimes it sticks. Yeah, it sticks to the RBD when it's in the up configuration, just as well as an ACE2 will stick to an RBD in up configuration. So if it goes up and down randomly, it has just as much a chance of sticking to your immune system as it does to a cell. So what's the point? It can't be chance, otherwise there's no point, no? It's not about knowing it's literal chance. You have hundreds if not thousands of interactions. I'm aware. Please read the first paragraph. I don't care. I don't know. I don't understand this. I don't have the background. I don't know this. I can't make sense of it. What am I reading this for? It makes use of a densely glycosylated spike protein to gain entry into host cells. The S protein is a trimeric class 1 fusion protein that exists in a metastable prefusion conformation that undergoes a substantial structural rearrangement to fuse the viral membrane with the host cell membrane. That substantial structural rearrangement happens after the connection. That's after the connection. When the S1 subunit is cut away. That's what that's referring to, no? This process is triggered. Oh, yes. This is not about binding. This is after it's bound. I should just read the next sentence. No, I don't even know where it went. You're triggering me. This process is triggered when the S1 subunit binds to a host cell receptor. That's the ACE2 thing. Receptor binding destabilizes the prefusion trimer, resulting in a shedding of the S1 subunit and a transition of the S2 subunit to a stable post-fusion conformation. That's when the cell membrane and all that shit is absorbed with the viral membrane or the envelope in the membrane. To engage a host cell receptor, the receptor binding domain, RBD, of S1 undergoes hinge-like conformational movements that transiently hide or expose the determinants of receptor binding. That's that up-down shit. These two states are referred to as down. Oh my God, maybe it'll say it. These two states are referred to as down conformation and the up conformation, where down corresponds to the receptor inaccessible state and up corresponds to the receptor accessible state, which is thought to be less stable. Because of the dispensable because of the indispensable function of the S protein. It represents a target for antibody-mediated neutralization and characterization of the prefusion S structure would provide atomic level information to guide vaccine design and development. But it's not telling what that doesn't say what turns it up or down. Why would you? No, I don't care. This doesn't matter. I don't care. Make a subreddit post about it, okay?
The way you feel.
Oh my God, we don't need this. I don't need this. Let me finish. 4 minutes.
Proposal in 2015, they done this very similar.
What do you actually disagree on? Are you trying to correct something Destiny said? He's trying to make the argument. I'm not even arguing this furiously. I don't know that enough to care. I think he's trying to argue that the protease furin can cleave that S1 subunit to allow entry into the cell, but it also cleaves every single time when new viruses are being manufactured inside the cell because furin is located in the endoplasmic reticulum Golgi apparatus or whatever. So he's saying that furin will always cleave the RNA strands as they're being manufactured, right? But then he's also saying that they also cleave when the thing is coming. It cleaves it twice. It cleaves everywhere. Furin is like a dual slit experiment where furin is cleaving all parts of it at all points in time, both when the virus is being manufactured and when the virus is attached to the cell, okay? That's what he's saying. No, it doesn't cleave the RNA, it cleaves the proteins. My bad. I shouldn't say RNA, it's not the RNA, it would be the proteins. Okay. I don't actually know, though, and I don't care, but I don't know. But I don't think it matters. I think there's a level of granularity that's not relevant to anything that will come up in this debate. But maybe it will. We'll see.
Work together. All of a sudden, they're starting to do this separately. So, I mean, to me, this is a signal that they're no longer collaborating on these types of things. So, I mean, I haven't interviewed Ralph Barrick to ask him, like, when did you stop collaborating with WIV? But this could be a good question to ask him. Maybe at some point he'll provide an answer.
Maybe going off, I'll have more questions about this during rebuttals, but back to FAL1 and CCCG. So one of the reasons, like, it's a very interesting idea that this introduces a novel cleavage site that could be used in analyses. And then so that I think is very interesting. One question I have about that is, what is the scale of viral engineering going on? And the reason I ask that is I could understand like a RIFLIP analysis or just, basically like a simple gel analysis to say, okay, our culture has lost the furan cleavage site. But if you're doing this at a larger scale, if you've got 1,000 cultures going or you've done 1,500 or something like that, it seems unlikely that you do that versus sequencing, especially given the context that you said that they have high levels of sequence capability. So I want you to speculate or kind of compare in what sequences that FAL1 cleavage site would be valuable. What number of viruses would they have to be engineering for that to be the preferred method versus using, their in-house Illumina?
Jesus.
Yeah, it's a good question. And, well, I think for me, sequencing, they have to, like, pass along to some other group and have to wait. And, I mean, if you're sequencing, could also be a cost issue as well. Whereas if you can run a simple gel with a simple digest with, like, primers that you already have that can amplify this region and, like, quickly screen your colonies if you're doing a lot of experiments and you want to do them quickly, not have to pause every time for sequencing at each step, I think this could be useful. But of course, yeah, sequencing, like, this is for, like, quick screening, and sequencing can be the validating step to make sure that you're not having some sort of false positive. In terms of, like, the number of viral culture where this becomes kind of intangible or not feasible, yeah, I'm not sure. And they could have, like, this, if they're using multiple viruses for like, screening multiple viruses, and if they're engineering novel furin cleavage sites in multiple viruses, they could have had the same furin cleavage site in all of these viruses with the same kind of tracking beacon that they could quickly do analysis, like on a panel. Okay, which viruses had this furin cleavage site get deleted in which culture conditions, or which, like, they could have then iterated on the actual furin cleavage sites and have different restriction enzymes based on, like, the underlying nucleotides. Maybe they wouldn't have, like, RRAR and is one example, but then they would have K or RARR, and they would use a different restriction enzyme that's, like, for those nucleotides that would work with the RA, and then they would have multiple screens going in parallel. And it's just very easy to I think to run a bunch of gels for this screening and quickly have your result rather than have to wait for sequencing. But I mean, yeah, this is all speculation, so I mean, it's as good as my guess is as good as yours.
No, very useful. Okay, well, I think let's take a 5-minute break, and then Peter, do you want to go after 5 minutes? Okay. All right, everybody will return after 5 minutes. I'll leave the video running this time, and then I will stop it after Peter's speech to give our mid-break.
Thank you, Jerry.
Okay, so I stopped sharing. Is that good?
Yep, great.
All right.
Wow. And I thought I was going to get through both of these today. Good job, guys. You fucking wasted my life. Only 9 hours left. I'm proud of you guys. Jesus. Fuck. I've got like 5 paper links open there. What I opened this, but we're not even ready to look at a video like this. I have a feeling the conversation here is going to be substantially more fucking retarded though than anything that we've watched in any of these videos.
And it's not just China. And this is the theory, but the thing about the lab leak and why I actually want to deculture. You had Josh Rogan on, and he did an excellent job.
Really good job.
That guy's
amazing.
What he expertly shows is if this is true, the lab leak hypothesis is true. Yes, the Chinese covered it up. Guess what? The Chinese are liars, and they covered up SARS, and they've covered up basically everything that's come out of their country for the last like 30 years. That's not a shocker. The shocker is Dr. Anthony Fauci, 2017, reverses the ban on gain-of-function research, directs this funding to EcoHealth Alliance, Dr. Peter Daszak, who then gives it to the Wuhan Institute of Virology for the study of novel bat coronaviruses. And then when the pandemic turns around, we take Dr. Peter Daszak as the only American to join the World Health Organization's investigation team into whether coronavirus leaked from a lab. And on 60 Minutes, I forget who was interviewing him. She goes, well, wait, so how did you verify it? Did you just ask them? And he goes, well, what else can we do? Because he's also half British. And that's how he goes, well, what else can we do?
Okay, I don't even care. This guy's fucking retarded. Okay. Wait, the MIW, tell me if you agree with this. I'm reading this guy's. Okay, cleave 1. During virus assembly, S is cleaved into S1 and S2, allowing that spike part to move up and down and pre-activating it. Cleave 2, during binding, the spike is cleaved to reveal the part that merges the virus membrane into the cell membrane. Is that accurate? There's nothing in there for you. Here. Go get him. What are you looking for? Oh, look. Wow. There literally is nothing in here. Absolutely nothing. Now who's the retard? Now you look like an idiot. Now you look like a dumb fuck. Now she's just sitting there like she's trying to prove a point. Okay, am I done? Can I be done for today? What did Scott say for the first example of, or for the first 3 videos of this? Oh, he didn't offer an opinion here? I guess he was just cataloging both sides' arguments. The MIW responded to your question. Yes, but only that first cleavage is done by Furin and unique to SARS-CoV-2. The cleavage at entry is in SARS, and in SARS-CoV-2 it is done by a different protease, not Furin. Cool, okay. All right. I love you. I'm going to sleep. I can call and explain. I don't care that much. Make a subreddit post, and we'll all read it and love you for it. Get me the fuck out of here. Short pine, fold, laundry, long, bucket, burn, sting, to do, scheme, stir, racket, beat, itch, hustle, and blend. Things that you can do to a bread. God damn it. You can blend it, blend things, or not bread, whatever. Blend, stir, beat. Eggs? Folded eggs? That's like a thing, right? Holy shit, I'm a fucking god. As baking ingredients, okay. I think you could technically do all of these to eggs. Beat eggs, stir them, blend them in a blender, and fold. An omelet. That's what an omelet is, right? Okay. Lists. A to-do list. A burn list. No, I mean a short list. Laundry list. Bucket list. Boom! Easy. Easy. I'm itching for it. I'm longing for it. I'm pining for it. I burn. And then a scheme, a racket, a hustle, and a sting. Scams. Boom! Easy. All right, I'll be back tomorrow. Don't die.